Good morning, everyone. My name is Rushad Pavri, and I'm a group leader at the IMP in Vienna. And today I'd like to talk about a completed project which deals with our new findings, the new and exciting findings on the role of DNA replication in the genesis of chromosomal translocations in cancers. So our lab works with B lymphocytes, or B cells. And as we all know, the primary function of B cells is to produce antibodies, which we all need for a robust immune system, for long-term humoral immunity. And the thing is that B cells have a limited repertoire of the antibody-producing genes, which are called immunoglobulin genes. But we have a seemingly endless repertoire of antigens out there from pathogens and vaccines. And so the B cells need to be able to generate highly specific antibodies against potentially any antigen. And so what it does is that it modifies that limited repertoire of genes through a process called somatic hypermutation. And what that means is that it expresses an enzyme called activation-induced deaminase when it gets activated, and that happens when it sees antigen. And this process of hypermutation results in the mutation of the variable region of the antibody that binds to the target antigens. And this leads to antibodies of very high diversity. This diversity is then checked by a process called affinity selection. Essentially what you get are cycles of mutation and selection, essentially like a Darwinian-style clonal evolution. And this results in B cells which have higher and higher affinity for the target antigen. And this process essentially then is the basis for all long-term adaptive immunity against all the pathogens, infections, and also the vaccines. But the process of somatic hypermutation is random, and this leads to two undesirable outcomes. The first one is that mutations in the antibody genes can lead to autoimmunity, so you can have self-reactive antibodies. Now, the process of affinity selection should take care of that, but it's not foolproof, it's not absolutely leak-proof, and so some B cell clones do escape, and this can have, of course, deleterious effects. The other is cancer, and that's because AID can act at other genes as well. It doesn't only act at the antibody encoding immunoglobulin genes. AID can also target oncogenes such as MYC and BCL6, which are the major oncogenes in B cells, and this can lead to cellular transformation and lead to lymphomas. And as a matter of fact, most mature B cell cancers have signatures of AID activity, translocations, and it has been shown in many cases that this is actually a good driver for tumorigenesis. So our lab is interested in all of these processes, or rather the molecular mechanisms that drive these processes. But today I want to talk about our recent work where we have uncovered a new mechanism that drives chromosomal translocations that are found in these B cell tumors. So at the molecular level, this process, which we call antibody maturation, occurs at the immunoglobulin genes, and we work with mice. So this is the mouse immunoglobulin heavy chain locus, which encodes for the heavy chain of the antibody. As you all know, the antibodies have a variable region and a constant region, which are encoded by separate exons in the locus. So when the B cells are activated, when they see antigens for the first time, as I mentioned, this enzyme AID is expressed, and AID is going to make mutations in a transcription-dependent manner in the variable region, but also in these what are called switch recombination sequences that are upstream of the constant region exons that are shown as rectangles here. These mutations can get processed to DNA double-strand breaks, what I call DSPs. So these double-strand breaks can recombine in a process called class switch recombination, which gives you new antibody isotypes, which have very important effector functions in the immune response. Now, where does the genome instability come from? As I told you, AID can act at other genes, and one of its favorite targets happens to be the oncogene MYC, and so it can make mutations at MYC. And these mutations can get processed to give you DNA breaks. And occasionally, a DNA break at the immunoglobulin genes can recombine with the break in MYC, and you can therefore get a MYC-IGH translocation, which is a very common feature in many B cell cancers, which is also the translocation. We use it as a paradigm in our studies. And there is at least one cancer called Burkitt lymphoma, where MYC-IGH translocations are the marker of this particular tumor. So what is unknown is what causes these DNA double-strand breaks to come together? How are they brought together to bring about a translocation? This is one of the big open questions, and is one of the things that I will talk about at the end. By the end of this talk, you will see what we have found to contribute to this particular point. So we started this project some years ago because we knew from a previous publication of ours that the DNA replicative helicase, the MCM complex, was required for class switch recombination. And we showed that this was required to regulate a step following DNA break formation and probably at a step that required the long-range interaction of these DNA double-strand breaks in the two switch regions. So the MCM complex is a hexamer. It binds to DNA and marks origins of replication. It gets activated by phosphorylation in S phase, which leads to the activation of its helicase function. That results in the unwinding of DNA strands. And along with all these other proteins, many other proteins and DNA polymerases, leads to the initiation of DNA replication. So given the similarities between class switching and the translocation biogenesis pathways, we therefore asked, do the MCM proteins play a role in the genesis of these translocations? And before I go further, I really must thank my fantastic team that has worked for several years on this. This project has been led by Mihaela Pecheva, who was a grad student in my lab, and she's really done pretty much all the experiments that I will show you, and it's really an amazing body of work. And bioinformatically, there was a lot of help from Tobias Neumann, who was instrumental, along with Daniel at a later stage, to set up all the pipelines. This project involved a large number of bioinformatic workflows, and really these two have done a superb job in that. So I want to thank them very much. Now, our system we use is the B-cell line called CH12, a commonly used line because it undergoes very efficient class switching and also translocations. And we deplete MCM complexes, or rather specifically the MCM6 subunit, with RNAi, so with a short hep in RNA targeting it. And this is a Western blot just showing you that we deplete MCM proteins to about 70 to 80% of the wild type levels. And under these circumstances, the cells are proliferative and they have a normal cell cycle. We also did a gene expression analysis shown here as an RNA-seq, and with the exception of MCM6, which shows a very strong downregulation, as we expect, there is very little change in gene expression. We also did a protein analysis, so this is a nuclear mass spectrometry analysis, and what we found is that the depletion of MCM6 leads to a downregulation not just of MCM6 protein, but also of other members of the complex, suggesting that this depletion has resulted in a destabilization of the MCM complex itself. But we did not find too many other important proteins to be deregulated, so in this system, both at the gene expression level and at protein abundance level, there doesn't seem to be any major perturbation in the cell. We also knew that mutation, that is to say the targeting of the enzyme AID, both at MYC and at the heavy chain, the immunoglobulin heavy chain locus, was basically the same. It was unchanged upon the depletion of the MCM complex. So we knew that at the time that the defect in class switching that we had seen was not because of defects in mutagenesis. So it was something downstream. So for translocations, we use an assay, a nested PCR assay, where we're simply looking at the production of a new product, in this case a translocation, which can be picked up by this assay. And the assay results in several bands on an agarose gel. You sequence these bands, you excise them, sequence them, and then you can quantify the number of translocations per, in this case, 10 million cells. And what we found is a very strong and significant reduction of translocations when we depleted the MCM proteins. And this was, as I said, in CH12 cells, which is a B-cell line. But to show that this was not unique to this line, we also took primary splenic B cells from mice and did the same experiment and got the same result. So the next question was whether, because we know that the MCM proteins are required for activating the origins of replication, the next logical question was whether the translocation frequency had anything to do with the origin activity at MYC and IGH. So we map origin or measure origin activity by this assay called short nascent strand sequencing, or SNS-seq. And essentially what the assay does, it is enriches for the leading strands that are found at replication origins. And it does so by getting rid of the shorter Okazaki fragments of the lagging strand, and then also makes use of the fact that DNA replication initiates with an RNA primer. And so by using this step of lambda exonuclease digestion, one gets rid of all of the genomic DNA, because lambda exonuclease acts 5' to 3', but it cannot touch the leading strand, the nascent leading strand, because the RNA primer protects it. And in this way, you enrich for the nascent leading strand, and after that, you can process those samples and go ahead with a deep sequencing experiment or even by a qPCR analysis at your region of interest. And so at MYC and IGH, the data looks like this. This is the SNS-seq data. There are two origins in MYC. There's a major one just upstream of the promoter, and there's another one down in the gene body. And both of these are downregulated when you deplete MCM proteins. Similarly, and what we had shown in our previous paper, there are a strong density of origins of replication inside the switch regions of the immunoglobulin heavy chain, and these are also strongly downregulated upon MCM depletion. And when we look genome-wide, when we compared the origins, or what we call the initiation sites, from the SNS-seq data and asked how they overlap in the known translocation hotspots in B cells, we found that this overlap was very significant. So origins of replication that overlap with the translocation hotspot were more strongly downregulated upon MCM depletion than origins that were not in translocation hotspots. So all of this told us that there was a correlation between the activity of replication origins and the genesis of MYC-IGH translocations. But all of this was correlative. It wasn't really giving us any functional insight. And so we decided to perform an experiment to ask directly whether replication origin activity regulates this translocation event. And to do this, we deleted this major origin of replication lying upstream of MYC. And we did this with CRISPR in the CH12 lines. And we generated multiple clones, and we checked them for MYC expression, for the mutations by AID in this intronic region, which is the hotspot for somatic mutation, and also for class switching in the clones. And all of this was normal, so that was good. We could then go ahead with the translocation experiment. And so we did the same translocation PCR, and we found this remarkable downregulation of translocation frequency in both of these, what I call the MYC delta origin or the MYC delta ori clones, showing you that this origin was essentially directly contributing to the translocation of at least of MYC-IGH. So this then led to the next question: what step of the biogenesis pathway is replication origin activity actually involved in? And so we knew it wasn't the mutation. So the next step is the formation of DNA breaks, which is absolutely essential for any translocation. And so we asked, is this replication origin activity required for DNA break formation? Now, DNA break formation is not easy to measure. They are particularly at genes like MYC, where the AID activity is very low. So the formation of double-strand breaks is a relatively rare event, and there are not very good assays to really measure this very well. So it wasn't an experiment we could do. So we asked the question in a different way, and what we asked was that if double-strand breaks are limiting for translocation genesis, then we should rescue the translocation frequency by rescuing double-strand break formation. And we could do this by coming in exogenously with a double-strand break induced by Cas9 right at the translocation hotspot at MYC. So if this double-strand break will effectively bypass the double-strand break formation at MYC, and if that was limiting when we deleted this origin of replication, we should get back wild-type levels of translocation. And so we did that experiment, and to our complete surprise, we found absolutely no rescue at all. So you see that when you use a MYC guide RNA, you get a nice robust translocations in the wild type, even more than a control guide. But when we do the assay in the delta ori lines, we can't see any rescue at all. And this was not because the guide RNAs that were used here to make were coupled with Cas9 to give you a double-strand break. This was not because those guides were defective, because if we look for indels at the cutting site of those guides, they're actually very robust. At 60 to 80% of your cells were getting an indel. So suggesting that this defect in not being able to rescue the translocations was not because the double-strand break formation was defective. And what this tells us is that the double-strand break that we came in with is many, many orders of magnitude greater than any physiological AID-induced double-strand break. And the fact that we still could not rescue the translocation told us that this defect in translocation biogenesis was downstream of double-strand breaks, but also independent of the break frequency. And so effectively, we could tell that the defect did not lie at the break formation step, but rather something downstream of DNA break formation and also independent of break frequency. So since we had so far only focused on the MYC-IGH translocation, we also asked whether translocations globally were also affected when we deleted this origin of replication or if we deplete MCM proteins. And so we did this assay called LAM-HTGTS, which essentially measures from a given bait locus the translocations to that locus. So in our case, the bait locus is at MYC, and we introduce a DNA break with Cas9. So once again, you bypass break formation at your bait. You then allow the cells to grow for a couple of days, so they will accumulate translocations. And then through a series of steps, you can enrich for the locus of choice, in this case MYC. And you can sequence the resulting captured DNA. In this case, we did it with nanopore sequencing. And this allows you to span the region with long reads and capture all the translocation breakpoints. And so the data looks like this. So there are different ways of visualizing it. The easier way is the plot shown at the bottom, where we are plotting the translocations in the wild type versus the delta Ori clone. And you see that all of these translocations are essentially all shifted towards the wild type, telling you that all of them were downregulated when you lost that origin of replication. The genes that are highlighted here are some of the well-known AID target genes that we also know are translocation partners of MYC. The circus plot on top shows you the same result. You have all the chromosomes lying around here. The MYC is at chromosome 15, where the bait is, and all of these arcs tell you where each translocation lies. It's essentially a fold change plot. So what you see here, everything in blue, are the translocations where you had more translocations in the wild type. And if there was something in the green part, it would have meant that you had more translocations in the delta Ori line. And what this essentially shows you is what you see here. All of your translocations are in the blue part, which means that all of them were downregulated when you lost the origin of replication. And the same was seen when we depleted the MCM complex. And so this told us that the defect we were seeing was not just for MYC IGH translocations, but rather for all MYC translocations. And as an example of what the actual data looks like, here's the immunoglobulin heavy chain locus. And you can see in blue are all the controls, the wild types, and in this case, the control LAC-Z, which is the hairpin that we use to control for MCM6 depletion. And you can see if you compare the blue with the green, or here in case the blue with the red, that there's a strong downregulation of translocations in all cases. So that is our conclusion again from this part, a very important one, because we found here essentially a novel regulatory step in translocation biogenesis, which acts downstream of double-strand break formation and also very completely independently of the break frequency. So what is going on here? We know that now it's something downstream of double-strand breaks. So the next step would be, in our case, the synapsis of this double-strand break. So the two breaks have to come together in nuclear space, form a synapse, ligate, and give you a translocation. So it has been proposed that replication can occur in the context of hubs or factories. In essence, if you have two origins of replication in a genomic neighborhood, they could come together and form a factory, which essentially means that their origins are activated synchronously at the same time. And if that's the case, we thought that that could explain how double-strand breaks could get synapsed. So if you have two double-strand breaks on these neighboring chromosomes, if the origins in those loci fire synchronously in a hub, that would bring about the co-localization of these two DNA breaks. This hypothesis would give you certain postulates. So firstly, it would mean that the AID target genes should have similar replication timing in a wild-type cell. Then the MCM depleted cell should have altered the replication timing, at least of the AID target genes. The MYC delta Ori allele, so the allele that lost the origin of replication, should also have an altered replication timing. And finally, the proximity of MYC and IGH, or of MYC and any of its target genes, should also be affected if you disrupted the replication timing. So replication timing is measured by an assay called RepliSeq. What we do here is that you pulse the cells with BRDU, it gets incorporated. You then sort the early and late S-phase fractions, you purify it, you enrich for BRDU containing DNA with an immunoprecipitation step with an antibody, and then you can do either genomic libraries to make genome-wide RepliSeq libraries, or you can look at loci of interest with qPCR. So RepliSeq looks like this. You get early and late fractions, as you see over here, and you then create a log2 track where you have the early versus late. And this results in this very characteristic pattern that you see in any wild-type cell, where you have the early replication domains as positive values and the late replication domains as negative values. And the same is true in this particular control in the wild-type. Now, when we deplete MCM proteins, you get this very striking phenotype where you've essentially lost this demarcation of early and late replicating domains. It looks as if there is no such replication timing signature at all. So that's one clear conclusion here. In the MYC delta Ori lines, because we've only deleted a single origin at MYC, we didn't expect to see any global changes, and in fact there are none. So globally, replication timing in these clones is perfectly normal. But if you look at MYC itself, a very remarkable thing is seen. So MYC occurs, is normally early replicating. It's part of a very large, more than 1 megabase pair early replicating domain. But when we delete that origin of replication, you see that a new domain is created, and in the zoomed in view it's better to appreciate. You see that you have a late replicating domain that has been created, approximately 60 kilobases, centered at MYC, leading to certain important conclusions. Firstly, that MCM depletion globally deregulates replication timing. This origin of replication that we've deleted at MYC leads to a switch from early to late replication. And also the fact that a single origin was enough to create a very large new late replicating domain tells you that that origin that we deleted is probably also a replication enhancer. So we know that replication timing is also correlated with genome architecture, particularly with genome compartments. And so we did a Hi-C analysis to ask whether any of these perturbations in replication timing also affected genome architecture. And the short answer to that is that there is no such change. The replication timing disruption, particularly in the MCM complex depletion, where you have this complete loss of early and late replication domains, did not lead to any change in genome architecture, as you can see in these Hi-C matrices and zoomed in here lower. And of course, we never expected anything to happen in the delta Ri lines, and that's exactly what we saw. All of this now led to the final question whether replication timing was directly required for translocation genesis, because so far we had shown essentially a correlation between this disruption of early replication timing and the loss of translocation. So to directly address this, we asked whether you could rescue translocations by rescuing early replication at MYC. This is the wild type MYC, which has this origin upstream that we've deleted in the delta Ri line. So that's what I've already shown you. And so what we now did is we made a new line where we took this deleted origin, which is about 800 base pairs, and we put it downstream of MYC in a region that has no replication origin activity and no transcription. It's essentially an inert region. And we thought that if this sequence essentially acts autonomously within the locus as a replication enhancer, then it should restore early replication to this allele. And if that was sufficient for translocation biogenesis, you should also rescue the translocation frequency. And these are the two clones of the rescue. You can see that in the delta Ri line, there was that late replicating region, and that's completely rescued when you bring back the deleted region downstream of MYC. And most satisfyingly, this led to a pretty much complete rescue of MYC translocations genome-wide, not just MYC IGH, but everywhere. So these are the same plots here, where you can see that now you no longer see the scatter of plots only in the blue region. It's all more or less scattered around the boundary of the blue and the orange. It's probably easier to follow it over here, where you can see that you no longer see that scatter on one side as you saw it earlier, but rather it's more or less now along the diagonal. And so that means that replication timing was directly driving the genesis of these translocations. And so finally, we asked whether the nuclear proximity of MYC and IGH was altered when we disrupted replication timing. And we did this with a 3C qPCR assay, which is essentially a chromosome conformational capture technique, this time looking for interaction between two specific loci, two specific restriction fragments, one at IGH and one at MYC. And the results were as follows. So we found that the MYC IGH nuclear proximity was significantly lowered if you deplete MCM proteins. As a control, we also looked at an intergenic interaction in cis between MYC and its enhancer located further downstream in the PVT1 gene. And this was an important control and did not show any change, suggesting that the decrease was specific for these interchromosomal contacts. And when we did it in the delta Ri line, we saw something similar. So you see here the control, the delta Ri line has lower nuclear proximity, and this is rescued when we rescue origin activity by reinserting that origin downstream of MYC. And again, as a control, the MYC PVT1 interaction was not changed. So nuclear proximity was also affected by the disruption of replication timing. And finally, we asked, because everything so far was done in B cells in the context of AID-dependent translocation, and we were interested in asking also for our long-term projects whether this was a general mechanism for translocation biogenesis, not just in B cells and not just for AID-dependent translocations. And so we took a well-known translocation found in leukemia, in particular acute lymphoblastic leukemia, which is between the AF4 and MLL1 genes. And we do a similar translocation assay, except this time we guide RNA cutting once in MLL1 and once in AF4. And then with a PCR assay, we look for the translocation product. We did this with digital droplet PCR, where each dot over here is essentially a droplet with a PCR reaction. And this allows you to quantify things with much higher detail. And what you see here and what's quantified over here is that if you knock down MCM proteins, and these are two different hairpins targeting MCM6, you get a very strong loss of translocations of these MLL4, AF4 translocations. And when we did a repli-qPCR to look for replication timing, we found that AF4 and MLL1 were normally early replicating, but when you deplete MCM proteins, they start going late, particularly AF4 now becomes quite late replicating. And so once again, we could see that the loss of replication timing, or the disruption of replication timing, was correlating with the decrease in translocations, suggesting that replication timing driven translocation biogenesis may be a general feature of translocation biogenesis in across cell types and species. So I'd like to end with our summary and our model of how we think this happens, and I've already alluded to some of this. So we think that in translocations would occur preferentially between genes which happen to be in the chromosomal neighborhood to start with. And then in some of these cells, some of these cases, you would have synchronous origin firing between two active origins in these neighboring alleles, neighboring genes. This would lead to the formation of a hub or a cluster or a factory. And then if you have double-strand break formation in these two genes, the formation of the hub would essentially allow the double-strand breaks to be in close nuclear proximity, and this would drive their ligation and give you a translocation. But if, for whatever reason, in our case, the MCM depletion or the delta ori where we lost the origin of replication, one of the partners does not replicate at the same time, it's lost its early replication potential, then you would not have this synapsis occurring within the hub. The chances of the two DNA breaks coming together become much lower, no matter how high the break frequency is. And this would therefore then decrease the rate of translocation, as we've seen. So with that, I'd like to end and once again thank the fantastic team, Mihaela, Tobias, and Daniel, who really spearheaded this project in superb work lasting several years. Our collaborators for other projects that we're now working on with replication timing, and I did not get to talk about other exciting work in our lab on mechanisms of class switching and somatic hypermutation done by several people, also involving excellent collaborators. I would like to have a special thank you to our fantastic core facilities, without which we wouldn't be able to do what we do, our funders, and thank you for your attention.